Hello, everyone. Welcome to this Tuesday Research and Innovation Conference. Today, I'm excited to introduce Dr. Nirmal Banda, who's a research professor in the Division of Rheumatology, Department of Medicine. He'll be talking to us about stop-the-start therapeutic targeting complement 5 activating receptor to treat early rheumatoid arthritis and new pathway to precision medicine. Just a few notes on Dr. Banda. He got his Bachelor of Science in Biology at Punjab University in India, where he also completed a Master's in Physiology and Ecology, and then a subsequent Master in Physiology, Biostatistics, Research Methodology, and then a PhD in Epidemiology and Biostatistics. For the past 22 years, he's been working on the role of complement in anti-collagen antibody-induced arthritis, streptococcal cell wall-induced arthritis, and collagen-induced arthritis. That's impressive that a pulmonologist was able to say all that. But as he says, he's a complement junkie, so we're here tonight to have him. Welcome.
Thank you. Thank you. C5aR1 is a receptor belonging to the complement system. It is a 7 transmembrane G protein coupled receptor. Rheumatoid arthritis is an autoimmune disease, it's a lifelong autoimmune disease, and it can go away with the treatment, but normally it comes back. It is characterized by the presence of synovitis, cartilage damage, bone damage, followed by deformation of the joints. Early rheumatoid arthritis, by definition, when the symptoms onset less than 3 months, although this definition is subjective. These are my disclosures. This particular image shows the deformation of joints in arthritis as compared to the normal joints, and there is no deformation in normal joints. On the other hand, in knee joints from rheumatoid arthritis patients, there is complete destruction of the cartilage as compared to the normal joint with no rheumatoid arthritis. As I mentioned, rheumatoid arthritis is a autoimmune disease, but this disease is still manageable and there is no permanent cure for it. Although there are some generic as well as anti FDA approved drugs for the treatment of rheumatoid arthritis. For example, there are non-steroidal anti-inflammatory drugs, FOX2 inhibitors, corticosteroids and DMARDs, and also there is a new class of drugs known as biologics, and these are tumor necrosis factor alpha, and these work outside the cell. And there are inhibitors as well as monoclonal antibodies. One thing I like to point out, there's a drug known as anakinra or the Kineret. This is a FDA-approved drug for the treatment of rheumatoid arthritis as well as juvenile rheumatoid arthritis, and this drug was developed in our lab by Dr. William Allen, and I used to work with him, and I'm running his lab right now. And this drug is very successfully used for these two diseases. And this is L1 receptor antagonist. And then there is a new class of drugs known as JAK inhibitors, and all of these biologics as well as the JAK inhibitors, they do have serious side effects and also cause opportunistic infections and also various types of cancers, including lymphoma. Unfortunately, there is no drug based on the complement system, which is the focus of my presentation. Complement system is a part of the innate immune system. Overall, complement system gets activated by 3 different pathways, namely classical pathway, lectin pathway, and the alternate pathway. All of these pathways gets activated by different molecules. Regardless how these pathways gets activated, all of these pathways converge to cleave C3 into C3a and C3b. C3a binds to its own receptor known as C5aR1. On the other hand, C3b forms C5 convertase, which in turn cleave the mother molecule known as C5 and into C5a and C5b. C5a binds to its own receptor C5aR1, and also it binds to C5aR2. C5aR2 is a genomic twin of C5aR1, but it is deficient in G protein coupled receptor. On the other hand, C5b get deposited on the surface of cells and initiate the assembly of membrane attack complex to the cells of the bacteria. The major focus of my presentation today is on C5aR1 axis. There are some drugs based on C5aR axis, but these are not for the treatment of rheumatoid arthritis. So, for example, there is a drug known as aclazimab. This blocks anti-C5 and prevents the cleavage of C5 into C5a and C5b. This is very successfully being used for the treatment of proximal nocturnal hemoglobinuria, AHUS, as well as more recently myasthenia gravis. There is another drug which was approved by the FDA about 8 months ago, known as avacopan, and this is an oral anti-C5aR1 inhibitor. And this drug being used for the treatment of ANCA-associated vasculitis. There are a couple of inhibitors, for example, PMX53. This is another oral anti-C5aR1 inhibitor, and also aclazimab. These have been tried for the treatment of rheumatoid arthritis, but there was a complete failure in the clinical trial. The main reason for the complete failure in clinical trials are unknown, but there's a possibility that these drugs or the inhibitor they never reached to the knee joints or there was an issue with the half-life. And but above all, what we are thinking, these drugs have been used singly. Our hypothesis is that targeting both C5 and C5aR1 simultaneously will be superior to resisting anti-C5 or anti-C5a or anti-C5aR1 inhibitory antibodies or inhibitors. Pharmacological block of C5 alone cannot be total, and it is possible that the small amount of remaining likely will maintain the inflammatory state. Likewise, blockade of C5aR1 alone cannot be total, and competing C5a will activate some small percentage of C5aR1. By blocking both, we expect to synergistically decrease the possibility of activation. In order to provide the proof of principle that C5aR1 axis plays very important role in the development or pathogenesis of arthritis, we used a mouse model of arthritis known as collagen antibody-induced arthritis. This mouse model of arthritis is dependent on the complement system, and it is a very aggressive mouse model of arthritis. Basically, in this case, we inject a mixture of whole monoclonal antibodies into mice intraperitoneally, and these antibodies they directly bind to the cartilage and induce disease. At day 0, we inject a mixture of anti-collagen antibodies, and at day 3, we inject an injection of LPS to recycle the development of disease. And all mice, they start developing disease at day 4. And regardless of the study, we sacrifice these mice at day 10. On x-axis, we shows the days after the antibody injection, and on y-axis it shows the clinical disease score activity from 0 to 12. It's clear from the data that mice injected with PVS, all these mice develop disease. As I mentioned, the nature of this disease is aggressive and also is cumulative. On the other hand, mice injected with a single dose of 750 micrograms per mouse interperitoneally, there was more than 50% reduction in the clinical disease score activity as compared to the mice injected with PVS. These data clearly show that mice injected that C5 plays very important role in the pathogenesis of arthritis. We also induced disease in mice lacking C3aR1 or C5aR1. And we found out that there was more than 50% reduction in the clinical disease score activity in mice lacking C3aR1. On the other hand, mice lacking C5aR1, there was more than 90% reduction in the clinical disease score activity, indicating that C5aR plays much more important role as compared to C3aR1, which is upstream to the C5aR1. We also induced disease in mice lacking C6, which is a component, as I mentioned, of the membrane attack complex. And there was more than 50% reduction in the clinical disease score activity. Again, these data confirm that the terminal pathway of the complement system plays much more important role as compared to upstream complement receptor, for example, C5aR1. And also there is more than 50% reduction in the prevalence of disease in mice lacking C5aR1. We examined all data very carefully. In this case, in this particular study, all wild-type mice, as you can see, they were very aggressive disease. On the other hand, mice lacking C5aR1, basically these mice never developed the disease, and there was appearance and disappearance of the disease. All these mice were highly resistant to disease, and indicating that C5aR1 played a very important role in the pathogenesis of arthritis. We also induce disease in mice lacking the second receptor, C5aR2, which I mentioned is a genomic twin of C5aR1 and deficient in the G protein coupled receptor. And there was no difference in the clinical disease score activity in mice lacking C5aR2 as compared with the wild type. So we ruled out the possibility that this particular receptor plays no important role in the pathogenesis of arthritis. Since C5 is predominantly generated by the liver, and therefore we ask the question: Can we shut down the liver? Can we shut down the disease locally in the knee joint by shutting down C5 specifically in the liver? So we were collaborating with a company. And this company has already 4 FDA-approved drugs in the market right now, and they have a specialization to synthesize very good siRNA. So they synthesize C5 siRNAs, and these then were conjugated with the GalNAc. GalNAc can bind to ASPRD receptor, which is present on the surface or specifically on all hepatocytes. So we can deliver basically GalNAc conjugated C5 siRNA to all hepatocytes. In this way, we can shut down basically in humans just with 600 milligram per week of the dose of this particular GalNAc conjugated siRNA up to 13 months, more than a year. And therefore, we tested this in our model. So mice were injected, in this case, 3 times before the development of the disease. Mice were injected with GalNAc conjugated C5 siRNA, 5 milligram per kg or 10 milligram per kg. And also mice were injected with scrambled siRNA, which were also conjugated with the GalNAc. And as a positive control, we also injected mice with anti-C5 monoclonal antibody, 50 milligram per kg. And anti-C5 here we have used, this is BV5.1, which is the basis for ecology map. And mice were injected at minus day 5, minus day 3, and also minus day 1, followed by induction of disease. And all mice start developing disease. It's clear from the data that mice injected with either dose of C5, GalNAc conjugated C5 siRNA, there was more than 80% reduction in the clinical disease score activity. On the other, and it was similar to what we have seen with anti-C5 monoclonal antibody as compared to the mice injected with PVS or scrambled siRNA. So these data again confirm that C5 plays very important role in the pathogenesis of arthritis. And although C5, I have to mention, it works in an autocrine and paracrine fashion. So in this way, we can inhibit basically 99.5% of C5 in the liver for 13 months. Since the major focus of our studies is to develop a therapeutic for the treatment of rheumatoid arthritis or early rheumatoid arthritis, and then we examined early RA biopsies from 2 clinical trials. One was the pathobiology of arthritis, a cohort known as PEACE clinical trial, and the second was accelerating medicine partnership, also known as AMPS clinical trial. Both of these trials were conducted in the UK as well as in the US, and we also at this campus participated in the clinical trial. And the synovial biopsy, approximately 6 to 8 synovial biopsies, these were obtained from the wrist, knee joint, as well as the ankle from early rheumatoid arthritis patients. And then by using an ultrasound guided technology. And then these biopsies were divided, and the size of each biopsy was from 0.3 millimeter to 5 millimeter. And these biopsies were divided, and some were fixed in the RNA later buffer for single cell RNA sequencing analysis using 10x genomics, and some were fixed in the formalin for histopathology using hemotoxin and eosine, and also for various complement proteins using multispectral immunohistochemistry, and also quantitation using vector Polaris system. This particular slide shows the correlation between DAS28 ESR, which is a measure of clinical disease score activity in the clinic. And this is one of the approved criteria by the American College of Rheumatology. And the correlation was between DAS28 and synovial C5, means locally in the knee joint. And these results were surprising that in all 90, from all 90 patients in the knee joint biopsies, there was a negative correlation and between DAS28 and synovium C5 locally in the knee joint based on the adjusted p-values. We have taken care of all confounding factors due to the clinical nature of these studies. So these data are surprising and also interesting because C5 is the therapeutic target for many inflammatory diseases, including arthritis. On the other hand, in the paired sample, we found out there was no correlation between DAS28 and complement C5 gene expression in the blood. So these data clearly shows that rheumatoid arthritis is a local disease. It's not a systemic disease, just like lupus, and complement plays more important role locally as compared to systemically. And the top three line graphs show the correlation between DAS28 and synovial C5aR1 receptor, synovial C5aR receptor. So here in all 90 patients, we found out there was positive and significant correlation between DAS28 and C5aR1 receptor, and also DAS28 C5aR receptor. And these data are consistent. What I have shown you that mice lacking C5aR1 as well as C5aR1, these were protected from arthritis, although differentially. On the other hand, the third graph on the top, in the top line, that there was no correlation between DAS28 and C5aR2 gene expression, which is the genomic link I just mentioned. And again, these data are consistent with our mouse models of arthritis, where I have shown you that C5aR2 play no important role in the pathogenesis of arthritis because these mice were not protected, although C5a can bind to C5a equally. Bottom three line graph shows in the paired sample the correlation between DAS28 and among these receptors. There was no correlation. Again, it confirmed that complement receptors plays very important role locally as compared to systemically in the pathogenesis of arthritis. We examined synovial biopsies from early RA patients by using multispectral immunohistochemistry. Here we found out that there was a huge expression of C5aR1 in the synovial biopsies from early RA patients from 23 early RA biopsies as compared to C3R1 as well as CD21, also known as CR2. So these data again show that C5aR1 could be a very important marker, a biomarker at very early stage of the disease, and as compared to C3R1 as well as CD21, and could be a therapeutic target. And we also examined synovial biopsies from subjects with no history of arthritis and no history of osteoarthritis as well as opportunistic infection. And we found a minimum baseline of the receptors. These were present as expected, and we have used these biopsies as controls. This particular composite image shows the presence of C5aR, which is shown here in a cyan color by using a false color coding scheme using multispectral immunohistochemistry on the synovial lining. As you can see here, this is a very early stage of the arthritis because the synovial membrane cells are not dividing. So C5aR is predominantly present in the synovial lining. And we do believe that this particular disease initiates in the synovial membrane and also end up damaging the cartilage. C5aR is also present, which is shown here in a white color. One of the most interesting observations we have made on the section B, where we do see a huge percentage of cells, they were adipocytes or the fat cell, they are expressing C5aR on their surface. At present, we don't know what is the importance of C5aR on the adipocytes, but we do know that adipocytes, these are an integral part of the earlier synovium. So then we ask the question, which specific cells express high or low level of C5aR gene or the C5 gene expression? Here we found out that from the AMPS clinical studies, where we have analyzed data from 78 patients, although I'm not showing the data because the data is not available right now. In this case, we have used basically patients from all spectrum of the disease, early RA, ongoing disease, and differentiated RA. And we found out C5aR1 gene expression, which is highly expressed by the macrophages. On the other hand, C5aR1 is highly expressed by the macrophages. On the other hand, C5 gene is highly expressed both equally in the macrophages as well as in the fibroblasts. Then we have just published this data. Only two types of cells are present in the early RA synovium. Here, as you can see, as compared to the previous slide, the synovial lining cells have started dividing. The disease has initiated. Two types of cells are present predominantly. One are the macrophages, which are shown here in a red color. The second one, these are the fibroblasts, which are shown here in a orange color. So there must be a crosstalk in between these two types of cells to initiate the disease. Since the major focus is still on C5aR1 axis to find a therapeutic for the treatment of early RA, so we designed a conjugate known as antibody RNA conjugate. In this case, we have used anti-C5aR1 binding antibody and conjugated C5siRNA to the Fc part of the antibody by using a protamine linker. So we were able to conjugate 3 molecules of the protamine to the Fc part, and we were able to load 21 to 30 siRNA to each protamine molecule. The whole idea here is to use this conjugate as a GPS system. So anti-C5aR antibody that acts as a GPS system, once it's injected in the body, it will find all the cells expressing C5aR on their surface, and it will deliver the payload of C5 siRNA to cleave the mRNA so that it will impact the C5 protein as well as also the disease. So we did one experiment, and also here we injected this particular conjugate 3 times before the development of the disease, and we found out there was more than 80% reduction in the clinical disease score activity as compared to the control. So these data are very interesting, and here were consistent what we have seen with mice lacking C5aR1 receptor as well as mice lacking C5 or treated with monoclonal C5 monoclonal antibody, and there was also decrease in the prevalence of disease. So we have a patent on this molecule right now. I'm trying to license this particular molecule for the development, and so right now we are synthesizing a new conjugate which we call it version 2.0, which will be used as a drug for the treatment of early rheumatoid arthritis in preclinical setting, and we are generating a new anti-C5aR1 inhibitory antibody and humanize it. And then we will use the click chemistry to conjugate C5siRNA to the humanized anti-C5aR1 inhibitory antibody to generate stable antibody RNA complex with longer shelf life for therapeutic use. So click chemistry is basically a mixture of chemical reaction. These can be performed in one part and without any losing the product. And there is no consumption of oxygen and water in this case. And more importantly, the product remain in the same tube. And at the end, there is no need for any specific chromatograph method to isolate the product. And so the conjugate which will be generated, I'm showing at the bottom ARC version 2. So this is the putative model, and this whole process can be completed within 2 hours. And we have already published a couple of papers with our collaborator to provide the proof of principle that this technique basically works. And also the conjugate will be having a longer shelf life and also longer half life, and it will be non-immunogenic. So finally, conclusion, I just want to point out two high C5aR expression in the human syndrome indicate that it could be a valid diagnostic mark for early RA, therefore a valid therapeutic target. And then dual targeting of C5aR1 by using antibody RNA complex of humanized anti-C5aR1 antibody C5siRNA conjugate could be effective to treat early RA or establish RA and overcome the issues related to the failed clinical trials in RA using anti-C5aR1 inhibitors or anti-C5 antibody alone. And I would like to extend my sincere thanks to Dr. Marta Holler, Rheumatology Clinical Faculty, for providing early RS novel biopsies from two different clinical trials, and to Dr. Dimitri Simberg, Rob Simon, and Gaikwad for helping me in the clinical chemistry. They do have expertise in this particular area. And to Dr. Christialas from UK, he has provided me novel biopsies from early RA patient. Of course, to the NIH for the funding of this project for the last more than 22 years, and to Dr. Manish Chopra for giving me the opportunity to present this work in this particular conference. Thank you very much.
Thank you very much, Ivan. We have time for a couple of questions from the audience or online. Great talk, Banda. So I was, and this probably illustrates my profound ignorance of human histology, but in that picture of human tissue with the macrophage staining for C5aR, did you also stain for a general macrophage marker? I would have expected to see macrophages just percolating through the tissue.
Yes, you're correct. That this is a very early stage of the disease, basically where these are anti-CC positive, but these individuals, they have very low level of inflammation, and when we see under ultrasound, and so it's not a very advanced stage of the disease. Sometimes when I said less than 3 months is subjective. So sometimes there is only a single lining. You don't see the difference even with a normal joint as well as the early RS synovial. And sometimes, as you see, there are a synovial lining has 5 or 6 layers. So once this synovial lining start dividing, normally it's a 2-layer only. That's it. Not more than that. And in that slide, I have shown about 3 or 4 layers. So this means the cells started dividing. There is a hit already. So that's why sometimes there is neutrophils, macrophages, monocytes, they start infiltrating in the synovium as compared to the membrane. But the disease have already started there.
So basically, all the macrophages are expressing.
Oh yes, definitely yes. Okay. Yes.
Yes. So, given the central role of complement in innate immunity, would you predict that targeting C5 and the C5 receptor would make individuals more susceptible to bacterial infections?
Okay, this is a very good question. And the way I have designed this particular conjugate, what we are doing, we are not touching the entire complement system. So what we are doing, we are not touching C1q, we are not touching the phagocytosis, C3b, and we are also not touching the membrane attack complex. C5 is still doing its work. C3b is still doing its work. So all that phagocytosis as well as the membrane attack complex is contact. We are just using one side arm of the complement system because C5a is a chemoattractant which is highly pathogenic.
Right, exactly.
So that's we are leaving it. So no, by using this complex, we have very carefully designed, spent many years. So we are leaving the entire complement system intact.
Thank you, Irfan.